This method can help answer key questions in the alpha-synuclein field, such as elucidate the mechanism of aggregation, spreading, and toxicity of alpha-synuclein in vivo. The main advantage of this technique is that it does not require the time-consuming purification of alpha-synuclein-preformed fibrils.
Demonstrating the procedure will be Lucia Rota, a grad student from the Colla lab. To begin the procedure, prepare the homogenization buffer and keep it on ice. Next, homogenize the fresh or frozen tissue in a 1 to 10 volume of ice-cold homogenization buffer using a Teflon pestle homogenizer with 10 to 15 strokes. Afterward, transfer all of the initial homogenate to a microcentrifuge tube and centrifuge at 1000 times G for 10 minutes at 4 degrees Celsius in order to remove nuclei and unbroken cells in the resulting pellet. Subsequently, discard the result pellet. Next, transfer the supernatant to a clean microcentrifuge tube and centrifuge it at 10,000 times G for 20 minutes at 4 degrees Celsius using a refrigerated centrifuge in order to obtain the second supernatant and the pellet. Then transfer this supernatant to a polycarbonate bottle and centrifuge it at 100,000 times G for 1 hour at 4 degrees Celsius using an ultracentrifuge and a fixed angle rotor. Resuspend the P100 crude pellet with 500 microliters of the homogenization buffer.